So yeah, we might need an extension cord, but other than that, I think a longer extension cord, yeah, just to make sure we get the whole. Yeah, because there's no power on the wall, right?
Maybe we could just sit it on something else. I guess.
Yeah. No, I actually used that table the last time, and I got to pull that one out. I don't know that. Well, let's see how it goes.
Yeah, that's fine.
Yeah. What do you think? Yeah, I do. Yeah, I think it's too low now.
Okay, yeah, I made a presentation. That one I used
Yeah.
That's good.
I think it's better. Yeah.
Okay. He went to get a longer cord, so.
Yeah. Yeah, so this is the thing. I guess it says it's recording.
Is it?
Yeah.
So you can stop recording now.
So this, if you see yourself backwards, it won't be frontwards on that.
Jesus salvation.
All right, everybody. Thanks for coming. Good to see a full house in the summer. I'll just say hi to the people online, and maybe if somebody can give me a thumbs up online to make sure they can hear us okay. We are pleased to welcome Dr. Chloe Schmidt, who's visiting from the Center for Integrative Biodiversity Research. Yes. IDIV is the apparently the acronym in Leipzig. Chloe is a New Jersey native who went to Rutgers University and followed by a master's at the University of Lausanne in Switzerland, PhD in the University of Manitoba, and a postdoc at Yale with Walter Katz, and is now in Germany at IDIV. And we're really pleased to have her here today. She's going to talk about biodiversity and genetics and all sorts of things. Thank you.
So thanks for the invitation. I'm really excited to be here and visiting Halifax for the first time, and have enjoyed the chats I've had with you earlier today. And I'm looking forward to speaking more with everybody over the next couple of days. Today I'm going to be talking about the biogeography of genetic diversity. So we'll start just speaking generally about biodiversity. Biodiversity is multifaceted and hierarchical. And to get a better understanding of biodiversity and how it's maintained, and importantly, to be able to predict how it will change in response to human-driven global change, and hopefully to prevent and mitigate further biodiversity loss, it's really important to understand how different levels of biodiversity are interconnected and the extent to which there are kind of general rules governing the structure and patterns of biodiversity. A lot of what we know about biodiversity right now is about patterns of species level biodiversity, so specifically thinking about patterns of species richness at broad spatial extents. And while we know a lot about the patterns, the kind of underlying processes maintaining this diversity is still a little murky, and there are a lot of ideas, but no kind of general convergence about how generalizable these are across different places or taxonomic groups. And we also, in comparison, know very little about general patterns and processes involved with maintaining genetic biodiversity, despite the fact that genetic diversity is the most fundamental level of biodiversity. And it's super important for population resilience. So populations with higher genetic diversity tend to have higher levels of fitness, they have lower rates of inbreeding, and they are generally better capable of dealing with environmental changes through adaptive evolution. And so in this way, genetic diversity contributes to population's capacity to persist in the long term, which is then, of course, can scale up to affect the resilience of species level biodiversity, and as well as the resilience of communities, and maybe scaling all the way up to ecosystems. So my research program is really, the major focus is trying to get an idea of whether there is any kind of generalizable process that scales up from the population level to explain broader patterns of biodiversity across this hierarchy, so that we can have a more cohesive understanding about biodiversity and the processes that create and maintain it. And so I'm particularly interested in linking population level processes to broader macroecological patterns. And when thinking about these population processes, what exactly are we talking about here? So I like to think about these in terms of kind of very relatively straightforward and fundamental processes that are really generalizable across any kind of taxa. And so I think mostly about the interplay between population size and connectivity and relative strengths of different evolutionary processes that are acting in populations. And so I'll explain this a little bit because this will underlie everything I speak about for the rest of my talk. And so if we can think very basically about population size and how this affects microevolution in populations, we know from population genetic theory that larger populations tend to harbor higher genetic diversity. And part of this is because it's related to the strength of genetic drift working in populations, which is just random allele frequency changes over time. So small populations will have low genetic diversity and relatively stronger effects of genetic drift, which erodes genetic diversity. So that means small populations are losing genetic diversity more quickly than large ones, whereas in big populations where drift is relatively weak, we can maintain kind of more stable levels of genetic diversity for longer periods of time. And so these processes are also kind of indirectly related to natural selection and populations' adaptive potential, because when drift is very strong, so when allele frequencies are kind of bouncing around very randomly, that means that natural selection can't be very efficient in spreading beneficial alleles to facilitate adaptation. Whereas in these large populations where drift isn't so much of a factor, then natural selection can be very efficient and adaptation can proceed more easily. And so if we think about these processes working in an environmental context, so thinking about the ways that individuals and populations are interacting and moving around their landscapes, my research questions kind of focused around how we can link these basic population demographic processes to environments. So how do environments support different sizes of populations, different connectivity of populations, and how can we think about the ways that these affect genetic composition and microevolutionary processes acting on these populations, and then whether they scale up to produce broader patterns in macroecology. So in this talk, I'm going to try to cover a lot of ground. I hope I do that successfully. But we'll start at very big, broad questions at broad spatial scales, thinking about latitudinal gradients in biodiversity. And then moving on to how genetic diversity is distributed across biogeographic regions and species ranges. The goal here is to understand whether it's actually the case that population demography could be a contributing factor to biogeographic and macroecological patterns. And after that, we'll move into looking at how human-driven environmental changes is reshaping the organization of biodiversity and what that might mean for biodiversity at higher levels of organization. And this part will be mostly focused on the effects of urbanization. And so to briefly discuss macrogenetics, so these types of questions and this type of work now is falling under the purview of a field that's being called macrogenetics, which is essentially big population genetics. So it's asking population genetics questions at broad spatial, temporal, or taxonomic scales. And macrogenetics has really been picking up in the last few years because there's a lot of genetic data available right now. So evolutionary biology was amongst the earliest disciplines to really adopt a culture of open data and data sharing. And so, yeah, there's really just a lot of data floating around that has kind of yet to be capitalized on, but that's starting to be done more and more nowadays. And so during my PhD at the University of Manitoba, I started building a macrogenetic database of microsatellite genetic data. I started building it for terrestrial vertebrates. I started in the US and Canada, and I've since been expanding that with the hopes of getting all the data that's online. But that's been a long-term labor of love, and I'm still working on it now, very slowly. But to do this, I developed pipelines and tools for programmatically harvesting data from repositories like Dryad, which are now being used by some of my colleagues who are graduate students at the University of Manitoba who are helping to expand the database so we can cover more taxonomic groups. So at the moment, we've got over 4,000 locations, which are shown on this map here, for terrestrial vertebrates, as well as marine fishes and marine mammals. And each of the dots on this map represents a group of individuals of one species that was sampled at that location. And we've got around 350 species. And because we're working with the raw genotype data here, that means we can be really flexible in the types of genetic summary statistics that we're estimating from these data. And more importantly, we can estimate them in a consistent way across the entire data set, which helps to reduce some of the noise you would get in a typical meta-analysis framework where, you know, a lot of noise is added due to different methodologies and different metrics used. to estimate genetic diversity or differentiation or what have you. And so that's really a benefit of working with the data directly in this type of setting. Okay, so getting right into it, latitudinal biodiversity gradients. So the species richness latitudinal gradient is probably one of the best known patterns in macroecology. So this is where we tend to have higher species richness towards the tropics. And there have been, I think, over 30 hypotheses now trying to explain where this gradient came from, how it's being maintained. But there's still no real general consensus about what these processes are. And other levels of biodiversity have been found to have similar latitudinal gradients. So phylogenetic and functional diversity also tend to be higher towards the tropics. And it was generally thought that genetic diversity would also follow a similar pattern for various reasons. But yeah, we weren't sure about whether this was the case. And so this is kind of the first thing I wanted to study with these data. So on this map, we're not looking at kind of raw values of species richness. I used an approach with distance-based random eigenvector maps, which is just a method to pull out spatial variation in the data. And here, the colors on the map represent the predicted values of a regression using only this broad scale spatial variation in the eigenvector maps. And so we used this on to see if we could recover known stack patterns of species richness. And each of the sites here is a location where we have a genetic sample site. So we can see for the US and Canada that we've managed to capture the general patterns of species richness. This is for mammal data. So we've got a species richness hotspot here in the southwestern part of the continent. And when we do the same thing for genetic diversity, something very striking popped out. So we can see already looking at these two maps that it looks like they are not positively related, and actually they seem to be quite negatively correlated, which honestly is not what we were expecting to find, but it was super interesting to get this result. And we then later also tested it using data from amphibians, where we find generally the same thing going on. So especially along the East Coast here, we can see that in the species richness hotspot in the Southeast, for amphibian diversity in North America, we've got relatively lower genetic diversity in this area. And so obviously we wanted to dig into this a little bit further. And so we combed through the macroecological literature to find some hypotheses that were based on population level processes and population demography to see if we could try to build some links. And so the first one of these is the more individuals hypothesis. And this one simply states that areas with higher energy availability should be able to support more individuals, which means bigger populations and bigger communities. And neutral theory in ecology and population genetics predicts that diversity will tend to be higher in bigger populations and bigger communities. And so here we expected genetic diversity and species richness to be positively correlated. The second hypothesis is related to environmental heterogeneity. So this one suggests that areas that have greater environmental heterogeneity will have more different kinds of niches in areas, which means that we can kind of pack more specialized species and populations into an area of whatever size. But then because they're now specialized in these niches, they should have smaller population sizes that are potentially more differentiated. And so in this scenario, we expected that species richness and genetic diversity would be negatively correlated. And now these two hypotheses aren't mutually exclusive. So we could have an area with very high energy availability and also a lot of heterogeneity that might still be able to support bigger populations with higher diversity than a corresponding area with lower energy availability. And so we tested both hypotheses simultaneously using structural equation models, which is a nice framework to test network or hypothesis networks so we can have multiple response and predictor variables. And so I'll walk through our kind of conceptual model for what we saw was going on here. So first, population size should increase with increasing resource availability, which should in turn cause higher genetic diversity and higher species richness. And from the heterogeneity side, we predicted that heterogeneity would increase species richness and negatively affect genetic diversity. And our hypothesis is more or less supported. So this was the model, the final model that we have here. So just with a slight shifting of one of the arrows, we did generally find that both resource availability and heterogeneity seem capable of affecting genetic diversity and species richness. But of course, these two processes are kind of operating simultaneously. So it's a little more nuanced than, kind of saying they're only negatively correlated and it's due to heterogeneity. But it seems that with these kinds of simple and relatively coarse metrics that we used for resource availability and heterogeneity, we're able to explain a substantial amount of the variation in the data and do correspond with what we found in these maps. So this is already some pointers suggesting that we might be able to link population demography to macroecological patterns kind of well. And these maps and these kinds of ideas also lead to a lot of interesting questions about the geography of speciation, because we might expect in these areas with low genetic diversity, they also tend to be more genetically differentiated. Populations tend to be more genetically differentiated. So we can imagine that maybe over time, these differentiated populations are also, there's higher rates of speciation in these areas. But a lot of work done by others in this area seems to suggest that population differentiation isn't actually related to speciation rates. And so there's something potentially interesting going on here in terms of, what kinds of species or what kinds of environments support complete speciation, and how those macroevolutionary patterns can factor in to all of these questions. And just as a little teaser, because I know a lot of you work on marine, in the marine realm. So Eliana Caracaliu is a graduate student at University of Manitoba who I've been working with to build a database for marine fishes. And she's interested in asking a lot of similar questions about what are the types of environments and processes maintaining genetic diversity in the ocean. And generally finding it looks like there's higher genetic diversity in the Atlantic region, and there's some interesting relationships popping up with different measures of marine productivity and also human disturbance in the coastal regions closest to these sample sites. So stay tuned for those interesting results when they come out. Okay, so moving on to the second part of the talk here, where we'll zoom in a little bit further. This section is about, first, biogeographic regions, which are regions of the world that have distinct species assemblages. And our question here was to kind of ask why these still exist, why are they still detectable? So you could imagine that if populations are capable of adapting to new environments and expanding their ranges, then we should start to see the kind of barriers between these biogeographic regions start to homogenize more over time. And so thinking about this and thinking about potential limits to population expansion and adaptation at these biogeographic transition zones, we then borrowed another hypothesis from macroecology, and this is the rather well-known central marginal hypothesis, which was originally developed to explain why species have limited distributions. And the idea here is that populations should have their highest abundance in the core of their range, where the environment will be most suited to them. And as you move away from this range core, the environment will become less and less suitable, population sizes will get smaller and smaller until you reach a point where population is really no longer viable. And then that's where you get a species range that pops up. And thinking about this now in a biogeographic framework, if we think kind of roughly about a biogeographic region being a lot of species ranges stacked on top of one another, then this transitional zone between different regions is now an area that has a higher concentration of species range edges. And so therefore, it should be populated by kind of small populations where drift is acting very strong. So natural selection is really inefficient. It kind of creates this place where populations just aren't really good at adapting anymore. And so they're not capable of expanding into neighboring regions. And it's these contemporary population level processes that could be maintaining these boundaries that create patterns on biogeographic scales. And so to test this idea, I combined my data from North American mammals using metrics of population differentiation and effective population size, which tells us about the relative strengths of drift acting on populations. And then also beef this up with some other types of data. So the first one was the MacroPopGen database, which is another database of microsatellite genetic diversity with estimates of genetic diversity that were harvested from the literature. And here we've got coverage of mammals across the Americas, and then also a database of population density estimates for terrestrial vertebrates worldwide, but again, we've only used mammals across North and South America. And for each of these sample sites, we measured the distance to the nearest edge of a biogeographic region. And then we use Bayesian hierarchical models to test those effects of distance to the edge or position in the biogeographic range on our genetic measures of effective population size, diversity, differentiation, and population density. So I'm about to show you a plot of the model coefficients, but I first want to point out that this line is a zero effect line. So if our effect is substantially overlapping this, there's probably no effect. And effects that are on the right side of this graph will mean that the genetic or population density variable is higher further away from the edge of our region. And if it's on the left side, then it's higher closer to the edge of the region. And so quickly, the open circles here are the overall effect size of the effective distance across all of the species in the sample. It's flanked by 90–95% credible intervals. And then we've also included species as a random effect in these models to account for different levels of genetic diversity and different responses to the range position across species. So the bubbles behind these are species-specific effect sizes. And we can see for both effective population size and genetic diversity, we're seeing that in general, these variables tend to be higher the further away we are from the edge of the region. Genetic differentiation appears to be higher closer to the edges of regions. And we didn't really see anything going on for population density. So you can see here that the species-specific effect sizes are kind of all over the zero line. So there's really nothing generalizable we can say about this variable. And we think that could be because abundance tends to fluctuate on much shorter time scales than would these genetic variables. So all in all, what we're seeing here is that indeed populations closer to the edges of biogeographic regions tend to be smaller, drift tends to be stronger, genetic diversity is getting eroded more quickly, and they're more genetically differentiated, so potentially more isolated from populations that are closer to the cores of biogeographic regions, which suggests that it does seem to be that there's some kind of limit on adaptation in these areas that's maintaining the boundaries in these transitional zones between biogeographic regions. And I think this brings up really interesting questions about what those environments are, because if we think about it, these are regions that are limiting adaptation across hundreds of species at once. So if we really want to get this kind of more general understanding of the kinds of environments that affect genetic diversity and that scale up to species richness, this could be something that's potentially very useful for being able to predict the kinds of environments that will be important and to predict how changing these environments will change biodiversity patterns. Okay, so coming back to the central marginal hypothesis, the kind of clear next step here was to see whether this does hold at the level of species ranges. And this is a project that I am currently working on now at IDIV, and I finally managed to get all the data together for terrestrial mammals. This is what it looks like. And I basically did the same thing. So for each species, wherever they happen to be sampled, we measured the distance from the edge of each species range. And then again, we've used our hierarchical models to look at the effective range position on genetic composition. And again, we see kind of clear effects supporting this central marginal idea. So populations that are closer to the core of species ranges are bigger, they're more well connected, whereas populations closer to the edge are smaller, stronger drift, probably less good at adapting, and this is what eventually causes species range limits to emerge. And now the central marginal hypothesis has been really kind of debated in ecology. This is because a lot of the time when it's tested using abundance data, it isn't supported. And there's also a lot of different approaches you can use to test it. So for example, sometimes people measure the distance to the edge, sometimes it's the distance from the geographic center of a range, sometimes it's the distance from the niche center. And so we wanted to kind of get a better idea of what's going on at the genetic level and see if we could understand this a little bit. And so we took, for a few species where we have relatively good coverage of the range, we've mapped this population differentiation metric onto the range. And so the darker purple, bluish colors are very low differentiation. So populations are more well connected in these areas. And so we could kind of think of that as forming the range core. And we do look at population differentiation here. This is a principal coordinates analysis of a pairwise differentiation matrix. The colors match up here, so you can see that this central kind of cluster of population in this graph, all these are the least differentiated, and it seems to form a hub between the populations that are kind of further out in different directions. And so what we can see here is like the core of a species range can be quite big, and it might overlap the center of the species range, but we can see it also extends further up in different directions. And so again, there's just a little more nuance when we're thinking about kind of general ideas about core populations versus marginal ones. Okay, I'm not sure how long do we have time.
You are half an hour.
Perfect. Okay, so coming to the second half of the talk now, I'll discuss urbanization and how this is reshaping patterns of genetic diversity. And so urbanization is typically thought to reduce genetic diversity, and this is because we can imagine an urbanization gradient. We're going from large, continuous, nice natural habitats to ones that are increasingly fragmented and quite small once we get to a city setting. And so it's generally predicted that, again, as we're decreasing population sizes, drift is going to be stronger. And so we should end up in a situation in cities where drift is very strong, effective population sizes are low, genetic diversity is low. We're thinking about an urban matrix and how difficult it can be to move around different suitable patches. Populations might also be more isolated. And this isn't really great for a city setting because, as I mentioned in the beginning, having high genetic diversity and well-connected populations is really important for adaptive potential, especially in cities, to be able to cope with this really dynamic and kind of always changing environment. And so with this macrogenetic data set, we were really interested in testing to see whether this idea actually held and whether it's true across different kinds of taxonomic groups. And so we used mixed models again, and I'll just go through a cartoon of the results we found here. So any effects above the line would indicate that genetic diversity is increasing with urbanization, and below the line it's decreasing with urbanization. So for mammals, across all the species we tested, we saw that genetic diversity was consistently negatively affected by urbanization, as was predicted. For birds, on the other hand, we didn't find any average effect across all the species, but this seemed to be because we were finding some species responded positively to urbanization, whereas some species responded negatively. So overall, there was an average effect of 0. And then finally, in amphibians, we also found an average effect of 0, but then here it looks like the species-specific effects are also, there was really nothing going on in these data. And so this suggests that not all taxonomic groups respond to urbanization in the same way. And we think that this could be, across these taxa, at least related to dispersal capacity or movement ability in cities, because we could imagine that, birds can fly, so maybe some species are really good at maintaining large and well-connected populations in cities, which could explain these positive effects that we found. whereas amphibians are not super good at moving around and they're pretty sensitive to their environmental conditions. So we suspect that maybe populations in cities are even being wiped out before we're able to detect any effects on genetic diversity. And I'm currently working with a high school student who's doing a project to try to figure out what's going on with the bird data to see if there are any kinds of traits or traits of cities where these data were found that could mediate these different responses that we found. So she's already finding some cool things. It looks like birds like water birds or other shorebirds might have different effects from the rest of them. So looking forward to seeing what comes out of that. And so for the final research part I'll talk about here is related to neighborhood heterogeneity within cities. So a lot of work in urban ecology has mostly been done across the full urban to rural gradient. And at this stage, we're kind of ignoring the fact that cities are not homogeneous environments at all. And so we're just taking a picture of Halifax on Google here, but we can see that there's very clear differences in, the types of environments that we can find around a city and within the city. So we might have these downtown areas with a lot of buildings, a lot of urban infrastructure, there's parking areas, there's residential areas that might have a lot of street trees and yards and gardens. And thinking in terms of wildlife, of course, these environmental differences can have effects on population demography, even operating at a finer scale within cities. And the thing about city structure is that they're completely planned by humans, so we design cities. And so to some extent, they reflect the kind of different socioeconomic or cultural factors, particular to the city and whoever designed it. And so I looked into this with regards to the very long history of residential segregation of cities in the United States, particularly thinking about this policy of redlining that was practiced between the 30s and 60s at the federal level. So essentially, different neighborhoods within cities were given different grades based on their desirability. And then racial and ethnic minorities were systematically denied loans to purchase homes in better graded areas. And so we ended up with a situation in cities in the United States where the qualities, the environmental quality of these habitats of neighborhoods across cities is correlated with the racial composition of these neighborhoods in a process that kind of still lasts to this day, even though redlining was eventually outlawed in 1968 with the introduction of the Fair Housing Act. And so today, even still, areas that have received higher grades under the redlining policies tend to have higher proportions of white residents. They tend to be located further away from urban cores. There's more green space, more street trees, older canopy cover, whereas areas that received lower grades tend to be more located towards urban cores. They're more densely developed with urban infrastructure like buildings and roads, train tracks, highways, and they also tend to be more polluted. And so we predicted that these differences in the availability and connectivity of habitats across different environmental, different neighborhoods within cities could have impacts on population demography for wildlife. And due to this history of residential segregation, we would then expect the racial composition of these neighborhoods to be correlated with these genetic effects, with the potential demographic effects on wildlife populations. And I just want to emphasize that we're not only talking about the kind of availability of habitat in these areas in terms of different amounts of things like green space across different neighborhoods, but oftentimes there were physical barriers to dispersal introduced that could have specific impact on population differentiation. So for example, there are a lot of cases where actual walls were built between Black and white parts of towns, and also the highway system in the US does tend to be directly built through Black neighborhoods within cities. And so there are a lot of different reasons why we think that we should have detectable effects on wildlife demography. And to test these ideas, we whittled down the data set to only include populations that were sampled within cities based on US Census designation boundaries of urban areas. And here we're working with all 4 terrestrial vertebrate classes. We've got mammals, birds, amphibians, and reptiles. And then for each of the points on this map, we calculated the proportion of residents identifying as white within a couple buffer sizes around each of these sites, again, using census data. And then to test whether these sites were a good representation of the types of environmental differences we already know exist across the racial mosaic in US cities, we looked at the Human Footprint Index, which is a general measure of environmental disturbance, and human population density in relation to our metrics of residential segregation here. And we did find that sample locations with higher proportions of white residents tended to have a lower Human Footprint Index and lower human population density. And so, again, we threw this into a Bayesian mixed model, and we found that neighborhoods with higher proportions of white residents tended to have wildlife populations with higher effective population sizes, higher genetic diversity, and lower genetic differentiation, which does suggest that neighborhoods in cities with higher proportions of minority residents tend to harbor wildlife populations with, again, smaller population sizes, stronger drift, and potentially less capacity to cope with long-term changes in these types of areas. And something I want to point out here is that if you remember when I was talking about the taxonomic differences in responses to urbanization, when we're looking at the full rural to urban gradient, we can see here when we're dealing with wildlife populations that are only sampled in cities, we find that all taxonomic groups are overwhelmingly responding in the same directions. So there is this difference in scale when we're talking about working across the full urban to rural gradient versus when we're talking about intra-city habitat variation. And so here, you know, of course, this work does suggest wildlife populations living in minority areas might have a harder time coping with environmental changes. They might not be able to persist as long-term, but I think more importantly, this really points to the depth of the differences in environmental quality across different neighborhoods in the US that are correlated with race. And this kind of adds to a lot of evidence we already have, suggesting that they're, it's just a very clear issue for environmental justice. And of course, biodiversity and access to nature are really important for human mental health and well-being. And so building environments that can support wildlife in cities is therefore also beneficial for the human populations living in cities in the US. And so here, there's a lot of more work starting to be done looking at this variation in biodiversity data across different redline neighborhoods in the US, looking at the importance of urban parks in places that don't have a lot of parks. So these are very important kind of regions for sustaining biodiversity within cities. And in general, I tend to take a kind of optimistic view here, and that because cities are places that are designed by humans, that means if we are aware of these issues, we still have some chance of changing them for the better. And I think doing that is going to take actions that are as deliberate as the ones that kind of set up the situation in the first place. And of course, these problems are not unique to the United States. I use the US as a case study here because this practice of redlining was something that happened at the federal level, and it was applied consistently across cities nationwide. And so we thought that this would be a strongest case scenario for being able to detect effects. But of course, similar things are happening everywhere. And specifically in Canada, it seems like similar practices existed, but they tended to be more city or provincial specific. And so this would be something super interesting to work with sociologists or historians who are more familiar with the kind of differences in practices across the country to see the extent to which this holds in places outside the US. And so just to wrap things up here, I hope I've shown a pretty good case of how we can start to think about macroecological patterns from the population level. And I think these very fundamental microevolutionary processes that are associated with simple things like population size and connectivity can help us explain a great deal about the processes that maintain macroecological patterns and kinds of how we can understand biodiversity across the full hierarchy more cohesively. And this interface between macroecology and macrogenetics is, I think, a really exciting field of research that I'm super happy to be involved in right now. So whereas in macroecology, we tend to take a more pattern-focused and top-down approach to inference, with macrogenetics, we're really starting at the population and building up. So where they meet in the middle, I think, is just a really fruitful area of research to be in. And so I'll close here with just talking a little bit about some next steps and potential future directions in terms of this research. So I'm very interested in continuing to ask kind of basic science questions, but with conservation applications. So I didn't really get to talk here about the work that I started doing in conservation genetics and like trying to dabble a little more in policy, but genetic diversity is something that's underrepresented in global conservation policies, so thinking specifically about the Convention on Biological Diversity. Actually, it was only this past meeting where specifically genetic indicator got accepted. And so I'm really excited about using these data to think about how it can be useful for conservation and for assessing and monitoring genetic diversity in terms of developing things like indicators. I'm also got a lot of just kind of basic multi-species population genetics questions to be answered. So relating to intrinsic and extrinsic determinants of genetic diversity across species, thinking about population structure in a more generalizable context across species. So what in general causes populations to be more clustered versus more clinal, and can we use that for conservation applications? And of course, continuing to explore this realm of urbanization and human, social, and economic factors and how that can change biodiversity moving forward. And finally, continuing to ask questions about biodiversity change at the macro level. And I'll have a couple of master's students who will be starting in the next few months who are going to look at how we can relate population genetics to the magnitudes and directions of species range shifts, and also to variation in species abundances over time. And I would envision all of this kind of surrounding a kind of continued data synthesis effort to get all this genetic data together and nicely packaged in one place, with the goal of eventually the database becoming a resource that can be used by both researchers and also conservation practitioners. With that, I want to thank all of my wonderful collaborators that I've worked with over the years. Without them, I never would have gotten to where I am now. And also, of course, to all the authors who post their data and make it open for others to use, and to all of you for listening. So thank you.
Fantastic. Thanks so much, Chloe, for that. We'll open it up to questions for those online. If you can put any questions you have in the meeting group chat, I'll monitor them on my phone. But for now, anybody have any questions? Michelle, start here with Boris.
Fantastic talk. You're wonderful. And it relates to a lot of the interests of people here in the room. But also the city. So the city just planted a green network plan, which is to establish wildlife corridors and corridors for people moving between green spaces and making it accessible for everyone, human and non-human.
Yeah.
So I hope you can help with that. So my question really is how does the very types of regional processes you're looking at, like fragmentation, human impacts, the harvesting of population, all of that layered together, how is that affecting the macroecological pattern? It seems to me there's a little bit of a disconnect. You're looking at urban landscape and the effects of fragmentation and human effects on genetic structure, but all the macroecological stuff, including our own, still assume that sort of what we see at the large scale is sort of natural, but it's not, right? And so how does that play?
So do you mean kind of how? Yes.
How the regional process you're looking at for fragmentation or diminished populations, particularly from animals or populations can be fragmented, wouldn't that affect the overall macroecological pattern in a way that then deviates from what we would predict for just natural processes playing out?
I think it could. So thinking about that a little bit in this range edge work we're doing right now, thinking about how, again, these very kind of local and micro scale processes may change biodiversity. It seems like the kind of the main effects of the range edge is related to the steepness of environmental gradients. And so it seems like it's not super straightforward that, you know, populations toward the center that are bigger are going to be the ones to persist longer, because if you just throw down an urbanization gradient right in the middle there, you can cause this fragmentation that might lead to collapse throughout the whole range. And so, yeah, I think there is potential there to think that these, they're not very little, you know, a whole city isn't a small thing, but I guess the scale of macroecology, those types of disturbances can be super important and have ramifications, yeah, you know, throughout the species range and things like that. I'm not sure I entirely answered your question.
No, I agree. It's just an interesting field study, I think.
Yeah.
I think it's true.
Yeah.
So
yeah, and I mean, there's a lot more to be done, especially I think other environmental factors other than urbanization, particularly agriculture, which is like a major land use change, which I haven't looked into at all, but it's in the back of my mind. So yeah, there's lots of things to explore.
Yeah, it's a next presentation to it. Thank you. With regards to the first part, remind me if I'm correct, we describe this negative relationship between latitude and genetic diversity, right? Or
not quite latitude, but just species richness.
Species, yes,
yeah.
Species richness. And there was a pattern in California, southwest US, that showed lower genetic diversity. But it's also a very heterogeneous environment, and populations are quite distinguishable from each other. So I wonder, how do you deal with that aspect? Yeah, sure. I mean, populations are smaller, they have lower genetic diversity, but if you consider the species range in that area, then overall the genetic diversity would be high because there's genetic differentiation among populations.
Yeah, so that's thinking about, I guess, genetic diversity in if we think about genetic diversity of a species, yes, it's higher because you have a lot of distinct populations. Here we weren't quite thinking of it at that level. We're saying in general, just the population itself in this area will have lower genetic diversity related to heterogeneity. Okay. But yeah, so it's true that we can think about it at the species level, and you know how many different lineages or whatever you want to call them exist in a species. But if we're just thinking about the region and in general, populations tend to be smaller. Something interesting there is, of course, that's a very mountainous region, right? And so I'm already kind of working a little bit on looking at the same patterns across Europe, where, you know, in North America, we've got mountainous regions of heterogeneity that's parallel with patterns of precipitation and temperature. So there's a lot of kind of potential confounding environmental things going on there, whereas in Europe, since we've got the mountains that are kind of going horizontal, yeah, versus the climate varies differently. So that's something we're also interested to look at to see if we are able to reproduce these processes in other places. Yeah.
Other questions?
So, in a hypothetical scenario, if you were like trying to find a big city between cultures, would you like to choose a new urban area, or would you like to find older cities which are fragmented or doesn't have any big corridors? And if you choose the new urban area, what would your plans be for the urban densely populated urban areas with no environmental like green corridors?
Yeah, I think that's a super interesting question, but that would probably depend on what your goals are with biodiversity in the city. So, you know, it might be that in a very old city, it could be harder to bring back the wildlife where they've been absent for so long, rather than in a newer city, you might be able to kind of at least recapture what's still there and then help it build more. I think greening up any city is always for the best. It's just, you know, what kind of temporal scale we're thinking about this is going to work on. And in terms of where you would put green spaces, I think that's a very difficult question. You know, obviously they should be evenly dispersed around places, thinking about the types of species you have in your cities and their, you know, dispersal capacities. But that seems like a can of worms that I don't know if I'm qualified to get into, because, you know, there's all these economic relationships with where we're going to put green spaces, what happens when we add green spaces, because then we tend to gentrify areas and so rents go up, and so we're again excluding different types of people from these areas. And so, yeah, I think that's a really tough question. But, you know, in an ideal world, put green space everywhere you can fit it. So.
I do have another question within my head. For example, if you in a place where land is scarce, so you would have to have both biodiversity and farmlands. So what would be like? How would you like the concept of land sharing between farmlands and forest lands?
Mm-hmm.
How do you perceive that, or how sustainable is that?
You're asking all the tough questions. Yeah, I mean, they're already doing some of that, right? With putting, you know, hedgerows and things between fields to kind of maintain connectivity between forest habitats to get through fields. I can't tell you how well those work and for what types of species they work for, but I mean, that seems, I would say, a pretty good way to do it if you've got a corridor that's big enough to allow like larger species to go through, especially.
Tigers.
Yeah. Yeah, it also depends on where we're talking about what's happening for sure.
Thank you for your answers.
Yeah.
Okay, other questions from the room or online?
Thanks, I really enjoyed the talk. When you were talking in the first part of your talk, particularly in the central marginal hypothesis, I was really wondering what effect species traits have on these patterns of, you know, fixed population size and genetic diversity, these other things that we tested, and particularly body size and dispersal ability or mobility, right? And whether these are linked and whether you put those into your models to test. So I think we had random effects models where you, because I was trying to see from the level of icon silhouettes of the species whether it was a gradient of the species, and it seemed like there might have been in terms of body size.
So not actually. So I did do models with body size in them, and also the like direction of the nearest range edge, and there wasn't anything like obvious coming out. Something potentially interesting there was we found there's like a correlation between slopes and intercepts just for the population differentiation model, which suggests that species with higher levels of differentiation, so more population structure across the range, had these stronger central marginal kinds of patterns. But that wasn't obviously related to any specific trait. And we also accounted for phylogenetic relatedness in the models, and it was not doing a lot of work. So
is that surprising? Because you would imagine things that can move around more within the range would maybe have higher genetic connectivity.
Yeah, I think that's not surprising, but perhaps the kind of surprising part is that we didn't find that for any of the other models. Because then, like, if in general, this metric of differentiation tends to be correlated with genetic diversity, and so I would have expected to see a similar pattern for genetic diversity. And I'm still trying to think through why that's not the case. If the differentiation thing is maybe just a weird artifact of something happening in the model, but the the differentiation one is very intuitive and it makes a lot of sense. So I'm inclined to believe it, but yeah, still thinking on it.
Yeah. Yeah, but I saw the pattern of the effects on birds of urbanization. And we group up and group down, and you partially alluded to this, and you're still working on this, but you mentioned the ability to, what about I think, I forget what you mentioned, but the food, what about food choices, generalists versus, we look at that.
Maisha is looking at that right now. She's also looking at body size and dispersal capacity with the hand index. What immediately jumps to mind looking at the coefficients is like a split between the watery birds and not watery birds. So waterfowl and shore birds look like they're pretty negatively affected by urbanization, whereas everything else seems to be pretty okay. But yeah, she's still working through it right now, so I can't tell you what's happening with other types of factors, but I think that could, yeah, food could be a big one.
Yeah.
Just for someone who doesn't have a background in genetics, so I understand counting species, and that makes sense from my perspective.
Mm-hmm.
And then to quantify genetic diversity, I think you mostly use microsatellite markers, which are neutral markers, correct? Yes. So, like species diversity is not neutral, it's sort of adapted, and but you quantify genetic diversity. I'm just curious, like if you would use, say, whole genome diversity instead, would you find the same things, or when you would just look at adaptive genes, would you find the same thing in terms of gradients in genetic richness, or is some of what we found specific to neutral markers, specific to microsatellites? Has anyone looked at that?
No. So microsatellite, neutral microsatellites, in theory, are capturing genome diversity. Mm-hmm. So even if we were to use whole genomes, I wouldn't expect to find something different. The only reason we're not using whole genomes at this point is because there's not as many of them, and because they're more expensive. It usually tends to be like one sequence over here, one over there, and so we're not really getting a nice population level estimate. But yeah, I imagine if you use adaptive parts of the genome, you're going to find different things, and it's really going to depend on what species, what environment you're looking at, what the gene does. Most macrogenetics work at this point is done with mitochondrial DNA, assumed to be neutral, but they are genes mostly that we're using, and they have been finding quite different patterns. Yeah, and it seems for mammals and marine fishes and some amphibians a little bit that it is a parallel gradient with species richness, and we tend to have higher mitochondrial genetic diversity that is higher species richness. I think there could be something interesting in there thinking about like temperature adaptation, because mitochondrial genome is pretty in a lot of species. It's related to that metabolism and so I think that's a potential thing to explore from an adaptive diversity perspective.
All right. If there are no more questions, maybe we can thank Cole and anybody has any more questions, just send me an email. So,